Welcome to your Chapter 3 video lectures once again, where we are going to discuss Section 3.3 on enzymes. This will be where we are going to stop in Chapter 3. The rest of the material in Chapter 3, we are going to trickle into some of the other chapters of our book. So what are enzymes? I mentioned this in the previous section. They are catalysts that are going to accelerate normal physiological activities within our body. And they also, remember, help to decrease that activation energy of our cellular reactions. If our reaction is uncatalyzed, that means there is no enzyme present, and it might take a larger amount of activation energy in order for that chemical reaction to move forward. If a reaction is catalyzed, that means we have an enzyme present, which would decrease the activation energy for that chemical reaction to move forward. Enzymes are only going to facilitate reactions that would already take place, and they're going to increase the rate of product formation. Let's take a look at an exergonic reaction. We have our reactant here, which is sucrose, and this has a higher potential energy than the total potential energy of the products, which is glucose and fructose. Our activation energy is going to be required to initiate the reaction, and the presence of an enzyme is going to lower that required activation energy. Most of our enzymes are going to be globular proteins. They're going to range in size from small, being about 60 amino acids, to large, that could be 2,500 amino acids. And they have this unique three-dimensional structure in a protein chain called an active site. And this active site is where the substrate is going to bind, where we are going to form a temporary enzyme substrate complex. This active site is specific, so it's going to permit only a single substrate to bind to it, kind of like a lock and a key that fits in perfectly. And this helps to catalyze only one specific reaction. And here is a figure that shows us a decomposition reaction. So we could see here that we have lactose, and the lactose is going to be our substrate. So that attaches to the active site on our enzyme, forming that enzyme substrate complex, and the reaction is going to move forward. That bond is going to break and move into our products, which are released from that enzyme, and we have glucose and galactose as a result. And here is a figure that shows us a synthesis reaction. We are going to have our glucose molecules, which are monomers. We only have one of each. They are separate here. And they are going to fit into the active sites on this enzyme, forming our enzyme substrate complex once again. The enzyme is going to change its shape or its conformation, and this results in something called an induced fit between our substrate and our enzymes. Our bond is going to be formed between the new glucose molecule and the growing glycogen molecule, which we see pictured over here. The glycogen is released, and the enzyme now is free to bind to other substrates so that we can maybe add on more links to this glycogen molecule. Enzymes are named based on the name of the substrate or product, the subclass, and the suffix ase. So really quick, I want to talk about the subclass. There are 6 major classes of enzymes. I didn't put them on a slide for you because I don't necessarily want you to commit it to memory. You will learn more about this in other courses, but just in case you're curious, those classes are going to consist of oxidoreductase, transferase, hydrolase, isomerase, ligase, and lyase. And so notice how each of those classes end with the suffix ase as well. And so we just have some examples of some of these enzymes here, like pyruvate dehydrogenase, that's going to transfer a hydrogen from pyruvate, and DNA polymerase, that is going to help form DNA, and lactase that is going to digest lactose. So we'll talk about some of these enzymes as we move through the chapters.